The topic of my speak today is the silvery vitellogenin, we call VG, of leafhopper is an effector suppressing halogen peroxide in rice host. The outline of my talk as follows. The first part, I want to introduce the background of this research. The second, the second part are research content. Finally, we summarize the experiment result in this paper, and a simple conclusion of model is given. Let's move on the first part of this topic. The motivation of this work like this. Rice is the most important food crop in the world, especially in Asia, where rice is the staple food. However, disease and insect pest often occurred in the planting process of rice, which seriously affect the quantity and the yield of rice. Among the many kinds of insect pests in rice, leafhopper is the most serious. The leafhopper not only feed the rice leaf and shed, but also spread the rice disease, like RDV and RLP. Once rice is infected with the disease, it is difficult to recover, and it can cause the serious consequence of almost no production of the whole field. So the protection of plant against the insect attack is one of the important issues that need to study in agricultural application. Consequently, the scientific goal of this study is how to reduce the damage of insect rice. Vitellogenin, we call VG, is the uric protein crucial and essential nutrient or oxide development in female animal. In the last few decades, VG has been isolated and identified from a variety animals such as fish and bird, and from a variety of invertebrates such as crab. This study suggests that VG is a conserved yolk protein. Recently, VG has been detected in cellular protein of several animals, such as whitefly, aphid, mice, physalid, and plant hopper. We also collect the silvery with a special device and confirmed that there was a large amount of VG in silver of leafhopper by proteins. So let's ask a question. What is the function of the leafhopper silvery protein in VG in its feeding process? First, it was verified that VG can be released to rice host through silvery during feeding. We took 13 insects to feed rice and test them with specific antibody of VG. The result showed that compared with the non-feeding rice, there was a band of VG in feeding rice sample, indicate that VG can release into the rice. In order to move in intuitive observation lightning leafhopper silvery VG release, we used the fluorescent sleeves and immunofluorescence labeling of rice leaf after feeding. Confocal microscope observe the specific specific fluorescence signal in rice phenol, but not in fed rice. In the process of feeding, insects usually release salivary protein to cause the host plant defense response. In order to reduce the plant defense, insect involve effector to reduce the plant resistance and finally help the insect feeding. In order to further investigate whether the VG released by the salivary of leafhopper is involved in the defense response of the insect against the plant, we feed the rice with the leafhopper, inject GFP and the VG respectively. After the VG of leafhopper is knocked down by the RNAi, the rice was fed 12 hours to detect the insect resistance index of rice, include protein hydrogency and GA and SA. But the result showed that there was no significant difference in GA and SA metabiology levels between treatment group and control group, while the hydrogen peroxide level was significantly increased. Leafhopper treatment group compared with control feeding hole number increased significant, and the feeding hole produced a large amount of hydrogen peroxide deposit here. The result showed that treatment group had difficulty in feeding and was not conducive for feeding of leafhopper. The feeding behavior of leafhopper was measured by EPG here. Compared with the control, the number of protein A is increased. The time and the frequency of the S increased, and the time of active feeding decreased, and the number of feeding disordered wave F is increased. This result indicated that the treatment group was difficult to feed, which is not conducive to the feeding of the leafhopper. This result showed that VG released by salivary can reduce the resistance and help leafhopper feeding. Rice gene sulfite oxide, we call it SO, was identified by using VG in rice yeast library. SO is a key enzyme in sulfur metabolism pathway, which can oxide sulfite into sulfate and produce the hydrogen peroxide. We followed investigate whether VG affect the expression and the transgroup regulator capital of SO. We measured the SO in mice active sulfate ions construct and the rice after 12 hours of feeding, and the experiment that showed that the treatment group was significantly low and then the non-feed group. To further determine the interaction between VG and SO affecting the feeding behavior of leafhopper, we prepared the transgene rice, VGOE and SO knockdown. We measured the feeding transgene with the white type rice, SO enzyme activity and hydrogen peroxide concentration in the sulfide is increased. As expected, hydrogen peroxide concentration is well significantly reduced in the transgene rice. As CNXE is the anchor protein of SO. We can find the specific interaction between SO and CNXE by Y2H. Since Vg and CNXE and SO, there is an interaction relationship. So Vg and CNXE, whether there is a competition through in vitro protein competition. So we close the